This is video number 3 of gene expression. If you focus on the right-hand side of the screen here, to recap, we're talking about gene expression, which means making a protein. And it involves, in our cells, it involves 3 different parts, really a 4th we'll discuss later. But the first is transcription. That is using DNA, one template strand of DNA, to make your messenger RNA. This is the immature pre-mRNA. The second part is modifying the mRNA, and then we're now going to move on to using that mRNA in the process of translation to make a protein. Now that involves something called the genetic code. Don't mix up the genetic code with the human genome or all of the sequences within your body. The genetic code is how we read mRNA so that we can make proteins. I will show you in the next slide. You can read this part on your own. This is the genetic code. This is the translator from the RNA language to the protein language. The protein language is made up of amino acids. That is the monomer that makes up your proteins, and there are 20 of them. And you can see them listed here: phenylalanine, leucine, isoleucine. The RNA language is made up of codons. You will see that word in the previous slide, C-O-D-O-N-S. Codons are 3-letter words, 3 nucleotide words, and each word will dictate a particular amino acid. If you notice in here that we have what we call redundancy. Leucine, for instance, has 1, 2, 3, 4, 5, 6 different codons, which will all give me the same amino acid. That's called sometimes redundancy, sometimes degeneracy of the genetic code. This is a good thing, because if, for instance, I have a gene and it's supposed to read CUU, or the RNA is supposed to read CUU, but I have a mutation that leads to RNA that reads CUG, that third wobbly base is not important in that case. I still get the same amino acid. So it allows us to have mutations within certain portions of particularly the third base and not have any deleterious effects. A few other things I want to point out about this code is that there is one start codon. Every gene is going to begin with a methionine. It's going to begin with an AUG, and methionine will be called in to start the protein. That methionine is sometimes removed later, so not every protein actually starts with methionine once we're finished. And then there are 3 stop codons, UAA, UAG, and UGA. And we'll see what happens when we see those start and stop codons. So that is the genetic code. Let's go over a few of their features. Actually, can I go back a moment, because I want to mention something else. It probably shows up later, but I need to emphasize this now. The cool thing about this genetic code is that every single thing that lives reads the code the same way. So you're a bacterium, you say that these 3 U's is phenylalanine. If you're a plant, if you're a frog, if you're a human, you always read those bases as being the same amino acid. That's going to be really critical when we move into biotechnology, because we're going to ask bacterial cells to make human proteins. Okay, next. So let's take a look at this bacterial mRNA. You can see here is the mRNA, and here's a start codon, and here's a stop codon. Again, the mRNA strand is going to run from 5 on the left-hand side to the 3' end. We're going to read it in that direction as well. And then we're going to get a polypeptide. Remember, that's just another big word for protein. Also, let's define what we mean by the reading frame? So if, for instance, this is my mRNA that you saw in the previous slide, if you couldn't tell from that slide, but there are a lot of bases up here that we have in our mRNA, but are not actually going to be read as part of our protein. And what will happen is the machinery will look for an AUG. It'll scan for an AUG, the cell does. And once it sees the AUG, it says, okay, that's where this protein has to begin. And I'm going to read these codons in 3-letter words all the way down. So that's what's referred to as the reading frame. It's just 3 letters, move to the next 3, to the next 3, and on and on. If you have a mutation that leads to an addition, let's say, of a nucleotide, what happens is that the entire reading frame gets shifted. So instead of having CAG, we now have UCA. This G gets carried over with the C and the A of what should have been the second codon. So here's that GCA. And the very bad part about this is that once you insert that base, often every single amino acid, which is listed under here, every single one, almost every one, is messed up. Now, sometimes just by chance, it's not going to get messed up, but all of these are going to be incorrect. And this is something called a frame shift mutation. We'll discuss later. But we shifted the whole frame of reading codons because we added a base. Okay. Now let's take a look at the big picture here. Here's DNA. We pull that DNA apart. I use the 3' template strand, 3 to 5'. I made a strand of mRNA. And now here's that region that I said we don't read. We scan through, we look for an AUG, boom, that's my start codon. I am going to bring in a methionine because that's what the genetic code says for AUG. And I'm going to bring that amino acid in using another type of RNA, and the RNA is called tRNA. We're going to take a look at a picture of that in a moment. So what you can see is that it has these bases here, UAC, which are complementary to the bases on the codon part of the mRNA. Those amino acids will all be strung together, and then we hit a stop codon, and the whole machinery falls apart, and we now have this polypeptide or this protein, which can be folded up. The tRNA molecule, I apologize because I think my face is hiding part of that molecule. So let's use this one. tRNA is a single strand of RNA, but happens to be folded in on itself. And you'll see that bonding, hydrogen bonding between one part of the strand and another part of the strand. This is like what the hairpin loop looks like in the mRNA that I mentioned in termination of transcription. There are 2 parts to this. There is the part that holds the amino acid, that's the 3' end, and then there's this anticodon part. So the anticodon is the complement to the codon that you would find on the mRNA. Now, where does all this come together? What you can see here is we have an enzyme here, which is going to help to attach my amino acids onto the tRNAs. So the amino acid will sit in one site of this enzyme. I'm also going to use a little bit of ATP in here. And then in the third step, you can see this is where the tRNA comes in, and using the energy from the ATP, I am going to add the amino acid onto the tRNA molecule, and then the tRNA molecule is released from the enzyme, and it now is what we say called charged, which means it has the amino acid on it. Let's take a closer look at the ribosome. The ribosome is made up of two different types of molecules. It is made up of RNA, ribosomal RNA, and also it's made up of protein portions. And so you can see a closer up depiction of it here. It has two subunits, a larger unit here and a smaller unit here, kind of like a hamburger bun. And we have three different slots in here, if you will, called the EPA sites. We'll describe in the next video what happens in each one of them, but the E is where exit happens. The A is the A is over here, is the aminoacyl site. You don't have to remember that abbreviation. And the peptidyl site is in the center here. EPA, I always remember using Environmental Protection Agency, what the direction is. Okay, in the next video, we're going to take all of those things, the mRNA, the tRNA, and the ribosome, and show you how translation actually happens.